Assalamualaikum. We are going to discuss about topic 9, which is microorganism in soil for plant growth. Firstly, I want to introduction of Bacillus subtilis and plant growth. Microorganisms in the soil play an essential role in promoting plant growth by improving nutrient cycling, enhancing soil structure, and protecting plant from harmful pathogens. Among these beneficial microbes, Bacillus is one of the most important plant growth promoting bacteria by PGPB. This soil willing bacterium enhance plant development through several mechanisms, including nutrient solubilization, production of growth stimulation compounds, and suppression of plant disease. Bacillus subtilis is an aerobic bacterium with a rod-shaped cells. It is naturally transformable and extremely powerful genetic toolbox. It is growing fast and easy to cultivate. Bacillus subtilis be able to secrete hydrolytic enzymes directly and function as a plant growth promoter through production of specialized metabolism and other various factors.
Bacillus as a source of phytohormone for use in agriculture as a case study of microorganism in soil for plant growth, where the world population is expected to increase by 10 billion people in the year 2050, and this will become a challenge in agriculture industry to provide a livestock if it still practice a traditional method in food production and do not increase agricultural productivity for food demand in the future. Why bacteria? In this case, Bacillus is a bacteria genus with various species due to its ability that can act as a plant growth promoting microorganism, and it can increase nutrient uptake, and it can produce many useful compounds such as phytohormone, where phytohormone is essential component in plant for it to regulate all the psychological process, biological process, and exogenous interaction while being cheap for large production. Bacillus is a natural producer of phytohormone, where it uses the same microbiome metabolic pathway in a different environment for agriculture application, such as industry purpose. For instance, the extraction and purification of plant hormones are obtained from the growing bacillus in bioreactor tank through microbiome production. Meanwhile, in soil, the action mechanism of bacillus, where it naturally inhabits for it to interact closely with plant roots, including bacillus to detect root exudates, to colonize the root surface, and to activate its microbiome metabolic pathway. This natural mechanism allows bacillus to produce phytohormone for plant development and plant growth. These are the phytohormones produced by bacillus that plays a specific role in plant development and plant growth, such as auxins, cytokinins, gibberellins, abscisic acid, jasminic acids, and salicylic acids. Hence, these are result from using Bacillus subtilis in soybean plant for root growth promotion by auxins.
We're going to next case study, which is Bacillus amphibacillus as plant growth promoting bacteria in soybean and cannabis. On the article, we know major crop plants are challenged by biotic stressors, which include attacks from pathogens such as fungi, bacteria, and oxidative at the adverse stress such as radiation, salinity, flooding, drought, extreme temperature, pH, and heavy metals. These stressors have caused yield reduction in major crops planted throughout the world. Synthetic agrochemicals like a fertilizer and plastic used to combat the impact of stress are expensive, cause greenhouse gas emission, leaf chemical into the environment. Bacillus and plant growth promoting bacteria, PGPB. What is that? The need for sustainable food production is sustain a growing human population, call for alternative to agrochemical such as plant growth promoting bacteria PGPB that can be used to control biotic and mitigate abiotic stress. PGPB have the potential to support future sustainable agriculture crop production due to their relatively low impact on native soil, microorganisms, livestock, and human, and minimal impact on soil ecology and biodiversity. Their use in integrated pest management system has proven to be sustainable alternative to chemical plastic, definitely a safer, eco-friendly approach to managing plant disease. Alright, this is a mechanism of action of various PGPB. We can see what the process happening in that PGPB. We can see what the bacteria is involved in that progress. This is a proof. We can see in what the example is like a Bacillus varicella mode of action is inhibition of mobility of resistant microorganism and damage the particle cell wall through production of tangent like tomato.
Is there a biochemical pathway in the microorganism in soil for plant growth? Let's see.
Okay, firstly, potassium solubilization. Certain bacteria break down potassium containing minerals. They release potassium ions K+ into the soil. Potassium improve plant stress tolerance and enzyme function. Next, nitrogen fixation filament. Bacteria such as Rhizobium convert atmospheric nitrogen into ammonia and H3. This gives plant usable nitrogen for making protein and chlorophyll, and essential for growth, especially in legumes. And the last, phosphate solubilization. Microbes release acid and enzymes that dissolve insoluble phosphate. This increases the amount of phosphorus available for roots. Phosphorus is needed for strong roots and energy transfer. That's all from me.
Nitrification, microorganisms, nitrosomonas, and nitrobacter. The purpose, convert ammonia into nitrate, which is the form of the nitrogen most easily absorbed by plants. Next, sulfur oxidant. Microorganism, Cryobacillus. Purpose, convert sulfur into sulfate for plants uptake. And the last one, production of plant growth. Microorganism, Azospirillum, Rhizomonas, hormones production. That's all.